everyone. I welcome you all to the live problem solving session on NPTEL Bioinformatics Algorithms and Applications course. So this is live problem solving session for week 4. So in this session we will be discussing about the multiple sequence alignment followed by the phylogenetic tree construction. So in the previous classes we have started with the types of sequence alignment. So we have pairwise sequence alignment and multiple sequence alignment. Pairwise sequence alignment is the type of sequence alignment that involves aligning two sequences to identify the optimal pairing of the sequences. It is based on a scoring system that assigns a positive scores to the matching characters and negative scores to the mismatching characters or gaps. The main objective of pairwise sequence alignment is to obtain highest possible score, which indicates the degree of similarity between two sequences. So under pairwise sequence alignment we have discussed dot matrix, global alignment and local alignment. Then we have multiple sequence alignment, which involves aligning multiple, that is 3 or more biological sequences to achieve optimal sequence matching. These multiple sequence alignments are used to identify the conserved sequence regions and to construct phylogenetic trees, which helps us understand the functional and evolutionary relationship between different species or groups of organisms. In pairwise alignment, just for a recap, so we have discussed the dot matrix. So dot matrix method is also known as a dot plot method. It's a graphical method of sequence alignment that involves comparing two sequences by plotting them in a two-dimensional matrix. In a dot matrix, two sequences that must be compared are plotted against along the matrix horizontal and vertical axis. The method then scans each residue of the sequence to identify similarities with all residues in the other sequence. If a residue in one sequence matches a residue in another sequence, a dot is placed in the corresponding position in the matrix. Otherwise, a matrix position is left blank. If two sequences being compared are highly similar, the dot plot will display as a single line along the matrix main diagonal. However, when sequences are less similar, the dot plot will show more scattered plots with few diagonal lines, indicating that sequences will share less similarity. So dot plots can also used to find repeat elements in single sequence. Short parallel lines above and below the main diagonal indicate the presence of limits. We have also seen how a palindrome and an inversion will be identified using a dot plot. Then we have dynamic programming, so which is used to find the optimal alignment between two proteins or nucleic acids by comparing all possible pairs of characters in the sequences. So this dynamic programming can be used to produce both global and local alignment. So the global pairwise alignment uses dynamic programming is based on Needleman-Wunsch algorithm, while the dynamic programming in local alignment is based on Smith-Waterman algorithm. Followed by that, we have also discussed about the word or k-tuple based methods, that is FASTA and BLAST methods. The word method is a fast method for aligning two sequences. It begins by identifying the short identical sequences known as words or k-tuples and then uses dynamic programming to align these sequences based on the words. Now we have multiple sequence alignment. So multiple sequence alignment can be performed either using exhaustive or heuristic approaches. So exhaustive algorithms involve examining all possible alignments at once. So this includes a multi-dimensional search matrix which is required to perform multiple sequence alignment using exhaustive algorithm. Similarly, two-dimensional matrix is used in dynamic programming for pairwise alignment. This means that to align n sequences, n-dimensional matrix is required. For next, we have heuristic approaches that are typically used for larger datasets to achieve a more efficient alignment. So in heuristic algorithm, the first part is progressive method. So progressive methods are also known as tree-based algorithms, and these stepwise assembly of multiple alignments based on pairwise similarity. So this method is called progressive because it aligns sequences in a stepwise manner. So the first step is it performs a pairwise alignment of all sequences using a Needleman-Wunsch global alignment method and records the similarity scores. Then it converts the similarity scores into evolutionary distances to create a distance matrix. A guide tree is constructed from the distance matrix uh, using a guide tree is constructed from the distance matrix using the neighbor joining method. So the guide tree is used to direct the realignment of the sequences based on their relative position on the tree, starting with the two most closely related sequences and adding the more distant sequences one at a time until all sequences are aligned. So we have cluster based methods such as cluster W, cluster omega and T coffee are two well known progressive alignment programs. So commonly used algorithms for multiple sequence alignment include progressive methods like cluster W, MAFT, T coffee, as well as iterative methods such as muscle, SciBlast. So each algorithm employs different strategies and heuristics to generate alignments, balancing the computational efficiency with the alignment quality. So here I have the first question, so which how many sequences are needed for multiple sequence alignment? To perform a multiple sequence alignment, we need three or more sequences. In multiple sequence alignment, what does a highly conserved region indicate? Yeah, so it identifies the low sequence variability. So before starting with the phylogenetic analysis, I would like to show how a progressive method and iterative method look. So as I mentioned, the commonly used algorithms for multiple sequence alignment include progressive methods like cluster W, MAFT, T coffee, as well as we have iterative methods such as muscle and SciBlast. So these each algorithm employs the different strategies and heuristics to generate the alignments, balancing the computational efficiency with the alignment quality. So first we have cluster omega. I'll copy paste the link. So this is the home page. So this starts with the introduction to cluster omega, which is for multiple sequence alignment. So it says cluster omega is a new multiple sequence alignment program that uses seeded guide trees and HMM profile techniques to generate alignments between three or more sequences. So here next they specify the input type. So the input can be given in GCG format, FASTA format or EMBL for nucleotides only. So the sequence type include protein, DNA and RNA. So actually I have used the example sequences. So this include 1, 2, alignment of 3 sequences. So which is like hemoglobin subunit from Homo sapiens, mouse and Caprahicus. can also decide upon the FASTA format whether we want to see the cluster format with the character counts or in a FASTA format. So I'm keeping a default. So this is trial 1. Submit. So this is the multiple sequence alignment. So the sequences are aligned. Further, individual pairwise alignments can be viewed. In a color code fashion. Followed by that we have guide tree. So this guide tree tells human and mouse sequences are closely identical than when we compared with the cat. Followed by that phylogenetic tree, which is our next concept. So here crystal omega comes under progressive method. Further we have iterative methods such as muscle. So muscle is also part of EBA website. So here the website tells us that muscle stands for multiple sequence comparison by log expectation. So muscle is claimed to achieve both better average accuracy and better speed than cluster based methods or T-coffee, that is progressive methods, depending upon the options of output. So this tool can align up to 500 sequences or a maximum 5. So similarly here again I'm using the example sequencer, that is similar sequences what we have aligned previously. So both the websites have similar output interface, providing the multiple sequence alignment followed by color coded alignments, phylogenetic tree, and individual alignment files can be downloaded from here. So just to summarize about multiple sequence alignment, so these are the fundamental technique in bioinformatics that are used to compare and analyze the similarities and differences among the multiple biological sequences such as DNA, RNA, and proteins. It involves aligning 3 or more sequences simultaneously, thereby highlighting the conserved regions and identifying the functional motifs and inferring evolutionary relationships among the sequences. The primary goal of multiple sequence alignment is to identify homologous positions across the sequences which are indicative of shared ancestry or functional significance. This alignment So this alignment helps in various biological analysis including protein structure prediction, function prediction, phylogenetic analysis and identification of functional domains. So these MSA algorithms typically aim to optimize several criteria including maximizing the sequence similarity and minimizing the number of gaps introduced in the alignment and considering the evolutionary models to infer most likely alignment given in a set of sequences. However, achieving a globally optimal alignment for multiple sequences is computationally challenging due to the complexity of the problem and especially considering the number of sequences that are available. So the commonly used algorithms for multiple sequence alignment include cluster, MAFT, and T-coffee under progressive category, and we have muscle cyblast under iterative category.
Excuse me, I have a query. Which tool should be used and when? Matlab, when to use Clustol Omega or when to use Muscle or Tea Coffee?
No, it's just all the tools are near the same. Now, it's just based on the time. If you are having higher number of sequences and if you are okay to compromise on accuracy, you can use iterative based methods. Or if you want higher accuracy, you can go progressive based methods. Progressive based methods are time taking and less accurate. More accurate. Iterative methods are faster but less accurate.
So both these methods, like you have given the example of Clustol Omega and Muscle. So both these are, they are iterative or, I mean,
Yeah, yeah. Cluster based methods and MAFT and T coffee based methods are progressive based methods. So they take more time than muscle and cyblast, which is iterative based methods. But the alignments are more accurate in terms of progressive based methods than iterative based methods. So cluster is a progressive based method, muscle is an iterative based method.
okay, thank you.
So next concept is phylogenetic trees, which are diagrams used to represent the evolutionary relationship among the a group of organisms or sequences, such as genes or proteins. These trees illustrate the evolutionary history or phylogeny by depicting the branch patterns that result from divergence from common ancestry. So these trees are fundamental tools in evolutionary biology, genetics, ecology, and other biological disciplines. So the main components of the phylogenetic tree are nodes. So nodes represent the hypothetical ancestors or common ancestors of the organisms being studied. So each node represents a point in evolutionary history where a lineage diverges into separate branches, and nodes are usually depicted as a point where the branches split. So here next we have branches. So these represent the lineages or evolutionary paths connecting the nodes. The length of a branch is often proportional to the amount of evolutionary change that has occurred since the divergence from the common ancestor. So longer branches typically indicate greater genetic divergence or variance or a longer evolutionary time. Then we have root. So the root of a tree, which is the bottommost part, represents the common ancestor of all the organisms or sequences studied in the analysis. It is usually placed at the bottom of the sequence and represents the oldest point in evolutionary history. Then we have tips, also known as terminal nodes or leaves, represents the extent or observed organisms or sequences in the analysis. So each tip corresponds to a specific species, individual organisms or sequence being studied. Then we have internal nodes. So internal nodes represent hypothetical ancestors that are not directly observed but are inferred based on the similarities and differences observed among the extant taxa. So these nodes represent the point of divergence in evolutionary history. As I mentioned, we have branch lengths, so which represents the amount of evolutionary change, such as genetic substitutions or sequence differences that has occurred along each branch. So the lengths are often measured in units such as substitutions per site or time units. In some cases, we also observe a outgroup. So this outgroup may be included in the phylogenetic analysis. So this outgroup is a taxon or a group of organisms that is known to be distantly related to the taxa being studied. So including an outgroup helps to root the tree, providing the reference point for inferring the direction of evolutionary change. So similar to multiple sequence alignment construction methods, we also have various methods to construct the phylogenetic trees, which include distance based methods, maximum parsimony methods, maximum likelihood, and Bayesian interference methods. So these methods rely on different models of evolution and assumption about evolutionary process to infer the most likely tree topology to be given for the input data. So we have types of trees. So two types of trees, that is rooted and unrooted trees, which are the two fundamental representations used in phylogenetics to depict the evolutionary relationships among the taxa. So here taxa means the organisms or the sequences that we are using. So the distinction lies in whether the common ancestor of all taxa is explicitly shown, then it is a rooted tree. If not shown, then it is a unrooted tree. In a rooted tree, a single node called root node represents the hypothetical common ancestor of all taxa included in the analysis. So the tree is typically placed at the base of a tree. The root is typically placed at the base of the tree, indicating the direction of evolutionary time with branches diverging from it. Rooted trees provide information about the both relationships among taxa and directionality of evolutionary change. And these trees are also often used when the evolutionary direction is known or can be inferred, such when an outgroup is included in the analysis. Followed by that, we have unrooted trees. In an unrooted tree, the common ancestor of all taxa is not explicitly shown. Instead, the tree shows only the relationship among the taxa themselves. So these unrooted trees consist of branching patterns representing the relationship among taxa without indicating the direction of evolutionary change. So these two trees, unrooted trees, are often preferred when the directionality of evolutionary change is uncertain, or when the focus is solely on understanding the relationship among taxa rather than inferring the ancestral states. So these unrooted trees do not provide much information about the direction of evolutionary time, but they are still valuable for visualizing the evolutionary relatedness among the taxa. So the choice of rooted or unrooted trees depends on the specific goals of the analysis and available information about the evolutionary history of the organism or sequences under study. Rooted trees are preferred when directionality of evolution is known or when the inclusion of an outgroup allows for rooting the tree. Unrooted trees are used when focus is solely on understanding the relationship among the taxa without making assumptions about the direction of evolutionary change. So both representations are important tools in phylogenetics and provide valuable insights to the evolutionary process happening. So for a given number of sequences that are analyzed, number of rooted trees were given by the top formula, that is 2n minus 3 factorial divided by 2 to the power of n minus 2 into n minus 2 factorial, and number of unrooted trees are given by 2n minus 5 factorial divided by 2 to the power of n minus 3 into n minus 3 factorial. So before moving on to calculating number of possible trees with a given nodes, so let's understand the factorial concept. So factorial, which is given n followed by exclamation mark, is a product of all positive integers less than or equal to n. So n factorial means product of all integers which are less than n up to the so 1 to n, it's a product. So kindly solve this. So what is the value of 2 factorial, 3 factorial, and 4 factorial and 5 factorial?
Uh, I have two more queries. So one is, you mentioned the term outgroup. So how can I select which organism is best for my data? Which organism to be selected for outgrouping is the is more appropriate?
Yeah. So mostly the organism which is completely unrelated, or the sequence which has less identity or similarity to our query or input, the sequence of our interest is commonly placed as a outgroup.
So that could be anything beyond means beyond a certain organism. For example, if I am comparing bacteria, so any of the archaea or the animal I can take as outgroup.
Yeah, the sequences from them you can take as a outgroup here.
So anyone will be okay.
Yeah, actually, if you want to know the evolutionary direction of evolutionary change, only the outgroup is selected. So generally, provided the given orthologous or paralogous sequences what you consider, the outgroup is by default the sequence which has least similarity or identity with our input sequence or sequence of our interest. So we select outgroup based on the protein or the nucleotide sequence of your interest. So which is distantly related to it, it's automatically is selected as an outgroup. Even within the bacteria also we have different types of evolutionary changes happening. So if you try to build them, always a outgroup is absurd. That
Okay, and another question that I have is, when you drew the phylogenetic tree A, B, C, D, E, so here A and B are more closely related. So instead of writing A, B, C, D, E, can I write B, A, C, D, E? That will be same, na?
Yeah, actually we'll see that how this trees and the tree notations will change. Yeah, it's same. We'll see how to construct a tree and how these nodes are placed together in coming up slides.
Okay, thank you.
Yeah, so as I mentioned, factorials are product of all positive integers less than or equal to the given value. So 2 factorial stands 2 into 1, 3 factorial is 3 into 2 into 1, 4 factorial is 4 into 3 into 2 into 1, and 5 factorial is 5 into 4 into 3 into 2 into 1. So 2 factorial value is 2, 3 factorial is 6, 4 factorial 24, and 5 factorial 120. So given the formula, how many rooted trees are possible with 3 nodes? So here I have given reference formula how to calculate rooted trees. Okay, so this is what a factorial mean. Okay, we want to three. So according to formula, I replace n with a value of 3. So this gives 2 into 3, divided minus 3 factorial divided by 2 to the power. So this gives 3 factorial divided by 2 to the power of 1 into 1 factorial. So which is 3 into 2 into 1 divided by 2 into 1. So 2 2. So if for a given 3 sequences, there are 3 possible rooted trees. So now how many rooted trees are possible with 3 nodes? Sorry, how many unrooted trees are possible with 3 nodes? I'll wait for a minute for others to answer. So when you substitute the value of n in a given formula for number of unrooted trees, so this is the formula. So this gives 1 factorial divided by 2 to the power of 0 by 0 factorial. So which is 1 into 1 into 1, which is 1. So remember 0 factorial is equals to 1. So why number of unrooted trees for a given 3 nodes is 1. So next methods for constructing a phylogenetic tree. So these methods aim to infer evolutionary relationship among a group of organisms or sequences based on observed data such as DNA, RNA, or protein sequences. So here I'll be explaining some of the commonly used methods. First one is neighbor joining. So neighbor joining is a distance-based method that constructs a tree by iteratively joining pairs of sequences that are mostly similar, that is, which have smallest pairwise uh, distances until a complete tree is formed. So it starts with a star-like tree where all the tags are connected to a single central node, and at each step it identifies a pair of sequences or a taxa with smallest pair distance change or less number of similarities among those sequences and merges them into a new node, that is, it makes them a group connected to a central node. So this process continuously grouping of these sequences continues iteratively and gradually builds a tree until all tags are connected. Then we have UPGMA method, so which is unweighted pair group method for with arithmetic mean. So this UPGMA method is also one of the distance based method that constructs a rooted tree from a distance matrix representing pairwise dissimilarities between the taxa. So it starts by clustering the taxa into the group based on their pairwise distances, then repeatedly merges the closest cluster until a complete tree is formed. The branch lengths in resulting tree represents the average distances between the taxa. Followed by that, we have some probabilistic based methods which are like maximum likelihood method, which estimates the parameters of a model of sequence evolution to find a tree that maximizes the likelihood of observing the given data. So this maximum likelihood method calculates the likelihood of observing a data under different tree topologies and searching for a tree with highest likelihood. So this maximum likelihood methods are computationally intensive because they generate all the possible tree topologies, then select the best one. Then we have maximum parsimony, which is a method that seeks to find the tree that requires fewest evolutionary changes or the simplest explanation to explain the observed data. So this maximum parsimony method constructs a tree that minimizes the total number of evolutionary events. So these evolutionary events represents the substitutions or mutations indels that happen in a sequence. These methods observe these changes which are required to explain the differences among the observed sequences. This maximum parsimony method search through a space of possible trees to find the tree one with the fewest inferred changes or which has less number of divergent events happening, and the one with the fewest inferred changes typically is considered as the final tree. So this uses algorithms like branch and bound or heuristic search algorithms to get the confirmation of all the trees. So each of these methods has its strengths and limitations, and the choice of the method depends on factors such as characteristics of the data, that is sequence length and substitution rate, computational resources, or specific goals of the analysis. It is common for researchers to use multiple methods and compare their results to gain more comprehensive understanding of the phylogenetic relations. So the first method is UPGMA. I'll start with, so which is unweighted pair group method with arithmetic mean. So this is a simpling clustering based method that assumes a constant rate of evolution, and it needs a distance matrix for the analyzed taxa that can be calculated from multiple sequence alignment. So given this is the sequence alignment for residues, a sequence length of 8 residues. So first construct all the possible pairwise alignment. So here I am considering the alignment of A with B is equivalent to alignment of B with A. So we have 6 different types of alignments possible. So the mismatches in these alignments are highlighted in bold. So in pairwise alignment of A and B we have one mismatch. In A and C we have 3 mismatches, In A and D we have 4, and in B and C we have 3, 2. B and D we have 3 mismatches, and C and D we have 1 mismatch. So now I wanted to calculate a distance matrix. So first I initiate the distance matrix with diagonal elements being zeros, because the number of mismatches between A to itself is 0 and B to itself is 0. So why the diagonal has been filled with zeros, followed by the distance between A and B is 1 mismatch. So the distance is score is 1, followed by, since we consider alignment of A with B is equivalent to alignment of B with A, so similar value is observed in the second cell as well. Followed by that I have made a matrix. So this matrix is a symmetric matrix. You see, you observe similar values on two sides of the diagonal. So this is how a distance matrix based on dissimilarities or mismatches has been constructed. Then so here I am taking some other example for neighbor joining method. So this neighbor joining method starts with a star like tree where all taxa are okay. So here first I made all the possible pairwise alignments of the given sequences, then I have counted for the mismatches. So in the first A and B alignment, if we observe, we have only mismatch. In the last position, in A we have C, in B we have G, so there is one mismatch. So similarly for all the remaining alignments, I have noted the number of mismatches that are observed. Then I have planned to make a matrix. So when we align A with A, there is zero number of mismatches. So along the diagonal, so this is how a distance matrix will be constructed. So initially when you generate all the possible pairwise alignments and count number of mismatches in each pairwise alignment that are part of this multiple sequence alignment. So first take A and B. So in A and B we have only one mismatch. So the score of A and B is 1, since we have only one mismatch here. So similarly I have filled. For example, if we consider number of mismatches between A and C, I have mentioned it as 3. So when we see the alignment of A and C, in last 3 positions we have TGC in A and CCG in A. It's a complete mismatch. So 3 places the values have been mismatched. So a value of 3 has been placed. So as I mentioned before, neighbor joining is also one of the distance based method that construct a tree iteratively, but it starts with a star like structure that you observe on the top left of the screen. So where all tags are connected to a single central node. Now at each step it identifies the pair of taxa with smallest pair distance. So now given this distance table, so what are the taxa that has smallest pair distance between them? So which two has smallest value? Yeah, so we have smallest value is 3, which is between A and B and D and E. So in this neighbor joining method, the distance from new node to each of the old node is calculated as on the basis of the distance between the old nodes and the difference of the distance of each of the nodes to the root. So here, when you have these two scenarios of having least distance, one can start with any pair of their interest. So A and B has been grouped into one common central node. Now this new central node is named as A and B. The new matrix has been constructed by calculating the distance of A and B with C, A and B with D, A and B with E. So this distance is calculated based on the distance of C to A, distance of C to B and distance of A to B. So this is the formula that is used to calculate a distance of the central node of A and B with the all other nodes, that is C, D, E. The updated matrix looks like here. So in the updated matrix here again we have two scenarios that has closest values, that is 2, that is central node of A and B with C has a distance of 2 and D again has distance of 2. Further we have grouped C with central node of A and B, forming a new central node ABC. Further we have re-updated this distance matrix by calculating the new distances of central node ABC with D and central node ABC with E.
So alternately, you could have also grouped E with AB instead of C. That would also have been the same scenario.
No,
Can we do that?
yeah, alternatively we could have started with considering D and D, then
No, you have already considered, you have already grouped A and B together. So now in the moving from second step to third step, so here you have grouped C with AB. So here I'm saying instead of grouping C, grouping A with AB can also go.
no, once AB has been grouped, further no more A and B are individually considered. Only this central node, the darker black part is considered.
In this matrix only, like C and D, this E, they both have two values, which both same values, which are least.
Yeah, so instead of grouping C with AB, one can start grouping D and D. They can do this because it's both the values are same, so either both the ways you can do the the final tree output will be the same, it won't change.
Okay, thank you.
So finally we have left with grouping D and D, forming a phylogenetic tree has observed. So a star like phylogenetic tree has been now made into this tree. So this is how a neighbor joining method has been used to identify the closest sequences. Now if we observe, A and B are close to each other, D and D are close to each other, followed by C being close to A and B. Further we have neighbor joining. Given multiple sequence alignment, a pairwise distance matrix will be calculated. So these are the steps how we gonna solve in next. So group two most closely related sequences. So in this, the two most closely related sequences is A and B. Just give me a minute. Then here again I started grouping D and E, which are further next to closely placed taxas. Now before moving on, the matrix will be re-updated by computing the distance between the new group, that is central node, and all other groups. So once a grouping or a central node is formed, we recalculate the distance matrix and take the next smallest distance to forward. So A and B has been grouped first, D and D has grouped next, then we observe C having closest distance with ABC. So the given the sequence, so initially we have not mentioned anything about the distances. So I have mentioned about outgroup creating a node. So because of the evolutionary distance between F and other sequences has been very high, the F has been placed at the bottom. So F is outgroup for all these sequences A, B, C, D, E. So this is how UPGMA and neighbor joining methods are used for on any multiple sequence alignment to construct a phylogenetic tree, which leads to evolutionary visualizing the evolutionary distance between them. So given nowadays all these multiple sequence alignment tools has given their automatic guide tree or phylogenetic tree generation. So this is a tree. Here in this alignment of three sequences, the hemoglobin subunit of CAPI is the outgroup because the mouse and human have high similarity. Further, apart from this, we have iterative methods. So
Excuse me, I have a doubt here. So previously, when you have shown the this formula, so here there you have subtracted the value of DAB also. In this formula, that is missing. Yeah, yeah, in that yeah,
yeah,
here.
this is one way to group. So we have different ways of grouping
Okay,
here.
this is different
Method, yeah, distance based
algorithm.
method, yeah. So without considering the root node, like without considering A and B here, just the distance between A to next node and B to next. This is other way of doing. So different methods consider different ways of calculating the distance between the central node and the. So here what I mean central node is a point, the central point where A and B, like the C, you can see A and B is very similar to C. So a evolutionary guide tree or a branch has been drawn in the center of A and B. So the distance is calculated from this center point to C. If you calculate the distance from this, if we consider this has a central node of ABC, the distance of this point to A and this point to C is same, like 1 plus 0.5 is A, sorry 1.5, and it's 1.5. So this will give the evolutionary distance between the two sequences. So the evolutionary distance between A and B is 0.5, A and C is 1.5.
How do you calculate this 1.5 value?
Yeah, so it's the value is from 3. So the distance between A to C is 3. So equally the distance has been split here, average distance. So 1.5 towards from central node, A will be 1.5 ahead and below 1.5 is C. but there are different other ways also to calculate this score, but this is only average distance based score. Given distance between the central node of A and B is 3, so half the 3 is 1.5. So if you sum up the distance between A to C or B to C, it should come 3. This is what this average scoring means.
Okay.
So that's all I have for today. So I'll start with a quiz. So this is match the following kind of a question. So what do they represent? So root, leaves, branches and branch length. Consider left side values as numericals and right side as alphabets like 1, 2, 3, 4, and evolutionary time is A, origin of evolution is B, current organisms is C, and relationship between organisms is D. I'm repeating again, in this match the following, consider the left side values as numericals and right side has alphabets. Yeah, okay, the answers here are root is origin of evolution, leaves are current organisms, branches are relationship between organisms, then branch length is, yeah. So as I answers has been mentioned, so root represents the origin of evolution, leaves are tells us the current organisms or denotes the current organisms, branches tells relationship, and branch length is evolutionary time. So the question is, write the Newick notation for the given trees. So here I have given two types of trees, one with the evolutionary distance and one without the evolutionary distance. So write the Newick notation for the given trees.
Excuse me, sir, what we have to do here?
Yeah, write the Newick notation. I'll explain. So, Newick format is a way of writing trees in text files using parentheses and commas. It is used in phylogenetic and mathematics, generally to represent graph trees with the edge length. So there is a format called Newick format in which we represent them as a text, these trees as a text. Whatever information we infer from these trees is being.
Thank you, sir.
So mostly the Newick notation will have parentheses and commas. Try to group the closest ones together. So start from any one corner and then start the grouping, then it will be easy. So first find which is the possible closest pair. So in my sense, I'll suggest start with B and C, so which are close. So put B and C, which is separated by comma, in one parenthesis. Then find this B and C is close to next what. So first, to start with Nuvik notation, group the closest one. So I'll group B and C. So this is how you present the closest one, parenthesis, and each separated by comma. Then this grouped B and C is close to A. So one more. Now finally the cluster of these three is close to the cluster that is formed by D and D. So this is the Nuvik rotation for the given tree. Yeah, either A can be placed on either any side of the B and C, or either this could be like this as well. So when you have the scales or evolutionary distance given, so you add them as shown. So the evolutionary scale or distance are measured just before the node name using a colon. The same tree, it has been represented along with the evolutionary distances. So this is the Nuvik format, how the graph based or a tree image is being represented as a text. I hope everybody is clear how these trees has been drawn.
Yes, sir. Thank you, sir.
Okay. Next we have, which is a true or false. So which of the following is untrue about the UPGMA method? That is, this method is simplest clustering based method is UPGMA, which builds a tree by sequential clustering method. Given distance matrix, UPGMA method starts grouping two taxa with the largest pairwise distances in the distance matrix, or the distances between the new composite taxa or central taxa and remaining taxa are calculated to create the reduced matrix. The grouping process is repeated and another newly reduced matrix is created. So which of this is false about UPGMA method? So the answers are distance between this new central taxon and the all remaining taxon are calculated to create a reduced matrix. So kindly observe the steps here. So the matrix has been reduced. So the central composite taxa and remaining taxa distance has been measured. So now the matrix of 5 by 5 is reduced to matrix of 4 by 5, 4 by 4. Then in next step 3 by 3. So, given in a distance matrix, did we start grouping two taxa with largest pairwise distance in the distance matrix, or did we go with the smallest pairwise distance in the distance matrix?
Smallest.
Yeah. So, this is untrue. So, in this grouping, we start with the smallest pairwise distance. Remaining steps are true for UPGMA method. So it's a sequential clustering method that we cluster 2, then we cluster third taxa, fourth, fifth. So it's a sequential clustering method. So when we have a central taxa, we calculate the distance of remaining taxas from this central taxa, and the grouping process is repeated, and then newly reduced matrix is created, and this is an iterative process that we see in UPGMA method. So here are the main steps that are involved in UPGMA method. First, calculate the pairwise distances between all sequences in the dataset. So this can be done by using any of the standard distance metrics, that is using Hamming distance or Jukes-Cantor distance. Then create a matrix to store the pairwise distances between all sequences. Then cluster two sequences with smallest distance to each other into single group, and calculate the distance between this group and each of the remaining sequences using arithmetic mean of their pairwise distances. Repeat step 3 until all sequences have been clustered into single group. So using this clustering information to construct a phylogenetic tree where the length of each branch is proportional to the evolutionary distance between two sequences or groups that it connects. Then a bifurcating branch point in a phylogenetic tree is known as so so nodes represent the hypothetical ancestors or common ancestors of the organism or the sequences being studied. Each node represents a point in evolutionary history where a lineage diverges into a separate branches. So nodes are usually depicted as points where branches split. So that is a bifurcating branch point. Split is called as a node. So given a peptide that is lead seq, so calculate the molecular weight of the peptide based on the provided individual amino acid molecular weights. before answering, kindly make a note of this how a peptide bond is formed. So when a peptide bond is formed in between two amino acids, the water molecule is released. So when you are calculating the molecular weight of the peptide, so add the individual amino acids weight, then subtract the water molecule weight into number of times the number of peptide bonds that are available. That is sum of amino acid weights minus number of peptide bonds into 18.
Sir, please kindly repeat.
Yeah, so here to calculate the molecular weight of a peptide, so initially sum of individual weights of all amino acids that are present in the peptide, that is weight of uh, leucine, weight of aspartic acid, weight of alanine. So individual sum minus number of peptide bonds present into 18, which is molecular weight of water. So in this total sum minus number of water molecules that have been removed. So number of peptide bonds is equals to number of water molecules removed. So number of peptide bonds into 18. Okay, that will solve now. So here I have written sum of all. So this sum will be equal to sum is equals to 870. Now minus 18 into which is water molecule and number of peptide bonds that we see. So peptide bond between L and D, peptide bond between E and A, peptide bond between A and D, yeah, 6 peptide bond between D and S, peptide bond between S and D, and peptide bond between E and Q. So we have 6 peptide bonds. So this will give 762. So similarly I have made
Sir,
one more question.
please give that slide once more, please. The one before. Calculation.
Okay.
Yes, sir. Thank you, sir.
So similarly I have made one more question for practice. So this is the molecular weight of peptide NPTEL is. So these are some random peptide names I have taken just for the practice. first find the sum, then subtract water molecule weight, which is number of into number of times peptide bond is observed. Okay, so the weight given for asparagine is 130, proline is 110, threonine is 115, glutamic acid is 145, and leucine is 125. So their sum come up to 625 minus, so number of peptide bonds 1 2 3 4, so 4 into 18, so which is 553. So given for practice purpose, I've given find number of rooted and unrooted phylogenetic trees that are possible using 6 different sequences. We have done this already, so this is for extra practice again. this is some really big number to calculate. So I have calculated for number of unrooted trees, so I have replaced 6 here. So 9 factorial divided by 2 to the power of 4 into 4 factorial, so you'll have 945 rooted trees. So similarly, try to calculate for other sequences. So for given 6 different possible things, we have 945 rooted trees and 105 unrooted trees.
Sir, kindly give the calculation slide, please. Thank you, sir.
So now given a pattern, so identify a sequence which matches the pattern. Yeah, yes, 945 is correct. So I have given here what does the pattern and the symbols in the pattern represent. So X represent any amino acid at that position. Square brackets represent any one of the embedded one. The curly braces represent any amino acid except the one that are embedded, and the repetition, the number of positions are repeated in parenthesis and the number. So since after K and P curly braces and 2, so this denotes that the two positions here K and P shouldn't repeat. So this is what this means. So accordingly, which of the option that matches? Any other answer? Here the numerical number 2 in parenthesis repeat tells repetitions of the condition just before it. So the condition before it is the current position shouldn't hold K or P. So in the two positions this condition should be valid. So the answer is D, because in first position either P, L, A has to be there. So all the options satisfy this. Next position is any amino acid, that should be possible. So in the first we have X, so there is no amino acid with the name X, so option A has been ruled out. So we have other three options B, C, D satisfying. Then we have G in the third position. Option C has P in the third position, so option C has been ruled out. Now we have two options, that is B and C, B and D. So given in the fourth and fifth positions, K and P should repeat, but B has KK. So option B has been ruled out. So finally we have option D, which is correct. So in the last class, I have just missed this concept of frequencies of amino acid that is calculated. So we have two types of frequencies of amino acid that is calculated. Okay, I'll do it again. So as I mentioned here, the square brackets tells any one of PLA. So either in the first place P should be there, L should be there, or A should be there. X means any amino acid in the second position. Third position, it is already by default it is given as G. So the curly braces tells that except K and P, any other amino acids could repeat in fourth as well as fifth position, because the repetitive symbol 2 has been given. And final position is Y. So of these conditions, which satisfy is D option. Because first position it has L, so either P, L or A could be possible. Second is any amino acid, so it is given A. Third is by default it has to be G. In the fourth and fifth positions, either K or P, any amino acid is allowed, so R, A has been given, and last it ends with Y. This is how the option D is correct. So in last class I missed this amino acid frequency. So we have weighted and unweighted amino acid frequencies. So in unweighted, we consider all the sequences are of same weight, and we calculate frequency of a particular amino acid at particular position is number of amino acid at that position divided by total number of sequences in which position I is present, that is without gap. Then we have weighted amino acid frequencies, where the weight value is given for each sequence, and we have a binary delta value which is either 0 or 1. So this delta is 1 if amino acid A is present at that position in sequence I, at position I in sequence K. So I have taken a example multiple sequence alignment of 5 sequences. So at position 1, let's count the repeats of the nucleotides. Sorry. So at position 1 we have 4A, 0C, 1G and T, and gaps are 0, but total number of aligned positions is without gaps. So gaps are not considered in aligned positions here. So we have 5 aligned positions. So similarly for all the remaining 5 positions, the count has been made. Then the frequencies, that is number of alanines divided by total number of aligned positions. So we have 4 alanines and divided by 5, which is number of aligned. The frequency of alanine at position 1 is 0.8. Similarly in sequence 2 we observe 0 alanines are there, so the frequency is 0. If we see in the fifth position, we have 3 alanines, that is in sequence 1, sequence 2 and sequence 3, and total number of aligned positions here without gaps is 5. So the frequency of alanine at this position is 3 divided by 5, that is 0.6. So this completely I have filled the matrix using this. This is how unweighted frequencies for a given multiple sequence alignment is calculated. Followed by this, we have some conservation metrics, that is entropy based conservation metric and variance based. So the entropy is matrix tells that summation of unweighted frequency at particular position for a particular amino acid into ln of the frequency value. So given in position 1, the frequencies are, let's note down, that is 0.8, 0, 0.2, 0. So we have only 2 values here, 0.8. In position 1, alanine is 0.8, glycine is 0.2. So frequency of alanine at position 1 into ln of frequency of alanine plus frequency of glycine at position 1 into ln of frequency of alanine at position 1. Remaining values have excluded because they are zeros. Multiplying some value with zeros again leads to 0, and so it doesn't affect these values with the removal. So at position 1 the entropy based conservation score is 0.5. So similarly I have calculated for other positions as well. So I'll upload this slide, do go through it. Even I have made uh, a sample example for calculating weighted frequencies of amino acids. So here I am considering random weights. These weights are given in random order. So for sequence 1 and sequence 2 it is 0.2, for sequence 3 it is 0.1, for sequence 4 it is 0.3, and sequence 5 it is 0.2. So then I'll apply the formula according to the weighted frequency table. For each nucleotide in every position has been calculated. So this is finally weighted frequencies for the 4 nucleotides. Yeah, so that's all from me. If you have any other questions, I can take now. I'll upload this sample problems. Do practice these. If no other questions, you guys are free to leave. Yeah. Thank you for attending. See you all in the next class. Okay. Uh, that I'm not sure, but we'll make sure that questions doesn't have larger calculations that are involved. But regarding the use of calculator in the exam, do mail it in the Q&A portal of NPTEL Swayam portal. The NPTEL team will reply to you.
Sir, I think screen calculator.
Yeah, so I'm not sure of it. I can let you know in the next class. Or if you want answer, you can just add a query in the NPTEL that Q&A portal. I think everybody can access that portal, right? I can see some of you posting questions there. So I can ask NPTEL people to reply. So it can reach to whole community of this bioinformatics course. But calculating this factorial of 9 higher numbers, mostly we'll try to avoid it in the exams. We'll be keeping the calculations to smaller numbers itself.
So the questions will be MCQ based, or means like we are seeing in assignments, there are some blanks also.
Yeah, questions will be of all the types. So we have multiple MCQs, we have fill in the blanks, we have numericals, just like whatever the question type you see in the assignments, similar question types will be provided in the exam as well.